Hi, so we're back and we're going to start talking about the juxtaglomerular apparatus, which helps in maintaining blood filtrate formation as well as blood pressure of the kidneys. There are three types of cells. On here you can see the macula densa cells labeled, and you see the granular cells labeled. And then right in this area, we have mesangial cells. Mesangial cells are kind of like the macrophages of the kidneys. And we talk very briefly about them, so I don't have them labeled because I don't really talk a lot about them. The granular cells are smooth muscle cells, and so as blood flows in, if blood is too, or if the blood flow is weak, there's not a lot of pressure behind it, that's going to stimulate the cells here of the afferent arteriole to start secreting renin. And renin activates the production of angiotensin 2. Angiotensin 2 then is going to increase blood pressure, bringing our blood pressure back to normal, allowing us to produce more filtrate like it's supposed to. So weak blood pressure typically means weak or less filtrate production. Whoa, I didn't even touch it and it went to that. So the macula densa then, and I'm going to go back just for a second, okay? The macula densa, which is right here, are associated with the distal convoluted tubule. And so as fluid is leaving the distal convoluted tubule, the amount of salt or sodium is going to be detected by the macula densa cells. And this is going to stimulate renin production as well. So if we have high amounts, or I should say high amounts of sodium, I'm sorry, I'm totally messing this up. If we have high amounts of sodium in our urine, typically water follows salt, and what's going to happen is you're going to be removing a lot of fluid from your blood. And so that's going to stimulate the granular, or send signals to the granular cells again to secrete renin, which will increase blood pressure. And it also, when we activate renin or produce renin, renin activation of angiotensin 2 stimulates the production of aldosterone, and aldosterone is our hormone that is our salt-loving hormone. So we're going to increase the amount of salt we bring back into our blood. That's a weird picture. Okay. What in the world? Okay. I don't know what happened there. That was weird. All right. So here's a video that talks about the nephron. And so I'm going to watch it with you.
The functional filtration unit is a microscopic structure called the nephron. Each kidney contains approximately 1.3 million nephrons. Each nephron consists of a living vessel and a renal tubule. The renal corpuscle is the broadest structure in the renal cortex, consisting of a pulmonary membrane called the glomerulus, and it's the water glomerular capsule, also known as Bowman's capsule. Blood vessels enter and exit the renal corpuscle when it's nostril full. Its vehicle is continuous with the renal tubule. The glomerular capsule has two layers. The base of the layer invites the nephron into the glomerulus. It is composed of specialized epithelial cells called chromocytes, which have multiple individuated nuclear processes called insets. The parietal layer is a thick fibrous epithelium that forms a capsule around the glomerulus. The capsule is placed right between these two layers. Water and phosphoric compounds are passed through a filtration membrane formed by three components of the glomerulus. Small pores and capillary epithelium called filtrations extensively coiled proximal convoluted tubule originates at the tubular pole of the renal corpuscle and is continuous with the nephron loop. Each U-shaped nephron loop, also known as the loop of Henle, consists of a descending limb that extends into the medulla and an ascending limb that returns to the cortex. The nephron loop is divided into thick and thin segments. The thick segments are lined with simple cuboidal epithelium, and the thin segments are lined with simple squamous epithelium. Beyond the ascending limb of the nephron loop, the renal tubule coils again to form the distal convoluted tubule. Several distal convoluted tubules drain into collecting ducts that pass through the medulla of the kidney. These merge to form papillary ducts that drain into a larger calyx. As fluid passes through the renal tubule, useful products move out of the tubular fluid and return to the blood. And waste products in the blood move into the tubular fluid, ultimately to be excreted in the urine. Two types of nephrons can be identified based on their location within the renal process. Cortical nephrons, which account for 85% of nephrons, have their glomerulus near the periphery of the cortex, and nephron loops in the outer medulla. The remaining nephrons are called juxtaglomerular nephrons because their glomeruli are located at the corticomedullary junction and have relatively long nephron loops that extend deep into the medulla. Blood from intraocular arteries enters the vascular flow of each glomerulus through an afferent arteriole. The juxtaglomerular apparatus is located at the point where the afferent arteriole enters the renal corpuscle. The juxtaglomerular apparatus has three parts. The macula densa, composed of specialized cells of the distal convoluted tubule, modified smooth muscle cells known as juxtaglomerular cells, and extraglomerular mesangial cells. The juxtaglomerular apparatus monitors blood sodium concentration and adjusts glomerular filtrate volume through neuronal feedback mechanisms. The efferent arteriole from each glomerulus enters a second capillary network. From all nephrons, a renal tubular capillary network surrounds the proximal and distal convoluted tubules. This capillary network drains into intraocular veins. In contrast, afferent arteriole is located from juxtaglomerular nephrons and rectangular is filled with vasovector that surround the descending and ascending limbs of the nephron. From the vasovector, blood also flows into intraocular veins.
So, how much blood is flowing through our kidneys at any given time? Close to a quarter of our blood is moving through our kidneys at any given time. We're producing filtrate continuously. So we're filtering our blood, cleaning our blood. Filtrate is what is produced at the glomerulus. And we're producing a lot of filtrate every day, but the majority of the filtrate is going to come back in through reabsorption. Once the fluid moves into the renal tubule, it's called tubular fluid. And then at the proximal end of the renal tubule, PCT, or proximal convoluted tubule, we're going to produce, or we're going to reabsorb the majority of our nutrients, and we'll get rid of more waste products that couldn't go through the filtration membrane. At the distal convoluted tubule, we'll get rid of any excess waste products that our blood has, and we are going to continue to reabsorb water and important nutrients. Along the nephron loop, we're going to be reabsorbing sodium and then water. And then we're also going to be getting rid of some sodium at different times. So we're constantly moving the flow of water typically towards our blood or back to our blood so we don't lose that much fluid. Once the fluid leaves the collecting ducts, it's termed urine. And urine has mostly water, waste products, nitrogenous waste products like urea. There's going to be some hormones, some very small protein, amino acids, minor amounts of other small molecules that are going to leave our body. And so here you have in the renal corpuscle, we have the filtration membrane, and so we produce filtrate there. Once it moves into the the renal tubule, it becomes tubular fluid. It's going to move down to the collecting ducts, and then we make urine. So the remaining material is urine, and urine is going to be very dilute or very concentrated depending on our hydration level. So we have filtration, reabsorption, and secretion in the production of urine. Filtration occurs first at the renal corpuscle. Reabsorption and secretion occur along the entire renal tubule, but the majority occurs at the proximal convoluted tubule, and this is because the proximal convoluted tubule has microvilli, and microvilli increase surface area for absorption. So what does the filtration membrane look like? It's composed of three major structures: the glomerular capillaries, the basement membrane, and then filtration slits. So this is kind of what you're looking at. You have the capillaries, you have the filtration pores called fenestrated pores of the capillaries. We have the basement membrane. Hold on one second. Sorry, I had a phone call. So we have the fenestrated pores of the capillaries, and then we have the basement membrane, and then we have the filtration slits produced by the podocytes that surround the capillaries. So the fenestrations are highly permeable to lots of materials. Large proteins won't get through but typically other materials can get through. I hate it when it does this to me. There we go. Nope. Hello. There we go. The basement membrane only allows small amounts of materials through, so it's much smaller, excludes more materials, and only allows typically smaller, positively charged molecules, very little protein, very little amino acids, very little glucose molecules, stuff like that is going to get through here. And then we have the podocytes. The podocytes are a little bit larger, so if it gets through that basement membrane, typically it's going to be able to get through the podocytes, but some things are going to be excluded from the filter, or from becoming part of the filtrate from this area as well, because it's very negatively charged. Podocytes are very negatively charged, and the pedicels, which are extensions from the podocyte, wrap around, so their negativity is going to keep large molecules, proteins, and larger anions from moving through and becoming part of that filtrate. We have those mesangial cells, which are your part of that juxtaglomerular apparatus, and their main function is going to be they're going to be getting rid of any potential pathogens as the filtrate moves, but also they have some contractile properties that helps in maintaining blood pressure of the kidney, of the nephrons. So there's your entire filtration membrane labeled. I don't usually ask you to identify structures of the filtration membrane, but so in a picture, but I may ask a question about that filtration membrane, so you do want to know it. And so here's what is going to be excluded from the filtrate. You're not going to have cells and typically large proteins, but very small proteins, tiny hormones maybe will get through. Very few, but some maybe amino acids that are